Hello everyone, and welcome to the second part of Basics of Cancer from Nurture Oncology. Today, we'll be covering a few topics: mutations and carcinogens, radiation, chemotherapy, and resistance in simplistic terms. So, as we all know, a gene consists of regulatory gene and coding gene. Mutations in the former may alter gene expression. That is, in the regulatory region may alter gene expression, while coding region mutations might affect the gene product. Now, these carcinogens, they are, most of them are basically mutagens. So several types of mutations, they include base substitutions, frame shift mutations, translocations. They are involved in accumulating over time to transform a normal cell into a cancer cell, but a single event of chromotripsis may underlie a small number of cancers as well. Now, let us come to the next portion. What are these carcinogens? Carcinogens basically can be categorized as radiation, chemicals, or infectious pathogens. Now, radiation can damage DNA directly or indirectly through the formation of reactive oxygen species. Three intermediate reactive oxygen species formed from the radiolysis of water are hydroxyl radical, hydrogen peroxide, and superoxide radical. Out of these, hydroxyl radical is one of the most reactive substances. Now, many carcinogens need to be metabolized to form an ultimate carcinogen that covalently binds to DNA, and many chemical carcinogens add functional groups covalently to DNA. For specific cancers, viruses and bacteria, they're infectious pathogens and they have been classified as carcinogens for specific cancers. For example, HPV, human papillomavirus, cervical cancer. This is a very common example, and many screening measures are in um, practice to prevent this. And now, patients with xeroderma pigmentosum, I'm citing this as an example. They have an inherited defect in nucleotide excision repair. So, this means like repair process, if they don't work well as such, they can lead to accumulation of mutations. So one another thing, repair processes not working. The example xeroderma pigmentosa. Now, many patients with hereditary non-polyposis colon cancer, they have a problem with mismatch repair. Types of chemotherapy, they can be classified into alkylating agents, antimetabolites, alkylating agents, antimetabolites, or organic drugs. I'll be explaining this further. Another problem is the development of drug resistance, which I'll be talking about at the end of this session. So let's get to each of these. Conventional therapies often induce DNA damage to trigger cell death, and their goals include inhibiting DNA synthesis in rapidly dividing cells or interfering with cell division. Now, this is what the principle of chemotherapy is. We have three main types: alkylating agents, antimetabolites, and organic drugs. From alkylating agents, coming to alkylating agents, they form DNA attacks by covalent bonds. Form DNA attacks by covalent bonds and damages DNA. Examples include chlorambucil, cyclophosphamide, etc. Now, platinum-based drugs are there. For example, cisplatin, they bind to DNA to form attacks, again causing damage that trigger apoptosis. They are effective but have significant side effects like kidney damage. Antimetabolites mimic natural molecules in disrupting DNA synthesis. An example include 5-fluorouracil. They basically inhibit DNA synthesis. Mm, the next category is organic drugs. Doxorubicin is an organic drug that inhibits topoisomerase 2. Blocking DNA replication is used in treating solid tumors but can cause cardiac damage. So, they're like organic molecules, and one example I've cited here is doxorubicin, which inhibits topoisomerase. There are plant alkaloids such as vincristine, vinblastin. What do? They prevent microtubule assembly. That is, as we all know, cell cycle, it requires microtubules to integrate and disintegrate as such. So either like assembly can be prevented or a microtubule which has already formed can be stabilized so that this cell replication is not effective and it doesn't take place. So vincristine and vinblastin prevents microtubule assembly while paclitaxel stabilizes microtubules. So vincristine, vinblastin are one category and paclitaxel is another. Radiation therapy is used by about 60% of cancer patients and is delivered by electron linear accelerators, producing reactive oxygen species uh, which damage DNA, inducing apoptosis. Their effectiveness depends on oxygen levels in tumors, and oxygen amplifies ROS effects. Oxygen amplifies the effects of ROS. Now, um, let us come to mutation resistance. Cells with the same tumor may mutate differently, leading to drug resistance. What are the drug resistance mechanisms? Drug resistance mechanisms. Some of you who have completed pharmacology might already know this. Cancer cells can resist drugs by increasing drug efflux or reducing intake or modifying drug targets. Either of these are possible. Now, P-glycoprotein transfers drugs out of cells, leading to multi-drug resistance. This is a well-known example. Mutation in specific transporters, polyp transporter for methotrexate can reduce drug influx, and gene amplification, that is dihydrofolate reductase gene for methotrexate resistance. Um, they they can be. I mean, this is cited as an example. Gene amplification increasing drug target molecules. Enhanced DNA repair mechanisms like increased alkyl transferase can confer drug resistance. Now, there are strategies targeting DNA repair pathways. A strategy where inhibiting DNA repair pathways are targeted. A strategy where inhibiting one gene is lethal only in the presence of another mutation. For example, PARP inhibitors for BRCA, one or two mutations. PARP enzymes repair DNA single strand breaks. So inhibiting PARP causes DNA repair defects. So this basically causes single strand break repair. So if this is inhibited, that basically causes what? Cell death eventually. By inhibiting PARP causes DNA repair defects. In tumors with BRCA one or two mutations, PARP inhibition is lethal. So healthy cells with functional BRCA1-2 can survive PARP inhibition, sparing non-tumor cells. This is the principle used over here in case of BRCA1-2. There is a trial called olaparib trial in which this was tested out. Sorry if the screen is not syncing on time. Mm. And they are shown to have fewer adverse effects as compared to traditional chemotherapies. That's all for today. Thank you everyone for your patient listening.